Autophagy, while often described as the body's internal recycling system, also plays a subtle yet critically important role in protecting telomeres, the repeating DNA sequences at the ends of chromosomes that act as a kind of molecular clock for cellular aging. Every time a cell divides, its telomeres shorten slightly, and when they become critically short, the cell either stops dividing or undergoes programmed death. Accelerated telomere shortening has been linked to premature aging, reduced tissue regeneration, and a host of age-related diseases. One of the primary drivers of telomere shortening is oxidative stress, reactive oxygen species, ROS, generated by normal metabolism, inflammation, or mitochondrial dysfunction can cause breaks or modifications in telomeric DNA, hastening their erosion far beyond what would occur through replication alone. This is where autophagy's role becomes profoundly protective. By clearing damaged mitochondria, one of the main sources of cellular ROS, autophagy reduces the steady, chronic oxidative insults that threaten telomere integrity. When autophagy functions well, the burden of oxidative stress on telomeres drops dramatically, allowing telomeres to maintain their length for more cell divisions and preserving the cell's capacity to regenerate tissues. Moreover, autophagy's sweeping removal of misfolded proteins and other cellular waste limits inflammation, another indirect but powerful contributor to oxidative damage. Chronic low-grade inflammation releases cytokines and free radicals that harm DNA, including telomeric regions, accelerating cellular aging. By keeping cells clean and minimizing unnecessary inflammatory signaling, autophagy creates a more favorable environment for telomere maintenance. Recent research suggests that autophagy might even influence telomere-associated proteins. These proteins form protective caps around telomeres, the sheltering complex, and regulate their stability and repair. Dysfunctional autophagy leads to increased cellular stress and a decline in the levels or function of these protective proteins, compromising telomere integrity. Meanwhile, robust autophagy helps preserve these protein systems, indirectly maintaining telomere architecture. What's exciting is that interventions that boost autophagy, like caloric restriction, intermittent fasting, regular exercise, and certain plant polyphenol, are associated with both reduced oxidative stress and slower telomere shortening in observational and animal studies. While causality in humans is still under investigation, these links highlight a hopeful possibility. By supporting autophagy, we may prolong the lifespan of our cells not just through general maintenance, but by safeguarding telomeres, the very structures that define their replicative potential. Ultimately, although autophagy doesn't directly rebuild telomeres, it powerfully supports their preservation by keeping the cellular environment free of excess oxidative stress and inflammation, key forces that drive telomere erosion. This indirect protection of telomeres is yet another way autophagy emerges as a cornerstone of healthy aging, helping our cells continue dividing and repairing tissues for as long as possible.